While DNA is replicated with each cell division, what does DNA actually do for cells? DNA contains the necessary instructions on how to build proteins for the cell and other parts of our body. There are actually several proteins throughout the body, such as hemoglobin, which helps blood hold on to oxygen for delivery, amylase, an enzyme that breaks down starch into simpler sugars, and melatonin, a hormone involved in your sleep-wake cycle.
Justin!
Huh? Oh, I'm up. What were we talking about?
It says here in the notes, transcription.
Okay, cool. Can I have some coffee? Yeah, gee, thanks.
Oh, sure. Something else it isn't my job. Hey, are we still rolling? Let's cut this.
Anyway. Well, besides hemoglobin, amylase enzymes, and melatonin, eye color is controlled by various pigments, but darker colors are due to melanin proteins in the iris. Eye color is determined at conception when mom and dad's chromosomes align to make you the zygote. The expression of melanin in eyes occurs after a couple of months after birth, unless the child is determined to have non-brown eyes. When a person with genes for brown eyes, and mind you, several genes control eye color, the DNA contains a sequence for making melanin. DNA houses the instructions for melanin, but for the melanin to be made as a protein, or for any protein to be made, DNA must undergo the process of transcription into a molecule called messenger RNA, or mRNA. And then mRNA is read to make a series of amino acids that are made by the ribosomes that build the specific protein melanin. That process is called translation, and we'll talk about that in a later video. The pathway that I just described is called the central dogma of molecular biology. That is, the steps needed to express DNA into proteins. DNA houses your genes, and the protein arrangement made is instructed by your DNA to make your phenotypes. There are two major steps in this central dogma: transcription and translation. In transcription, a specific sequence or section of DNA is accessed and copied onto a molecule called mRNA. In translation, mRNA is read by ribosomes to build a series of amino acids that eventually make that protein. This protein goes on to become the phenotype of a given trait. In this example, having brown eyes is the phenotype, which is a result of more melanin production in the iris cells. So let's recap. The setting for this example I will use in gene expression is eye color. A baby is getting ready to turn 2 months old, and their eyes are beginning to become darker. And this darkening process is due to melanin proteins. And in order for the iris to express melanin to make brown eyes, the DNA within these iris cells must be accessed. Remember that DNA is housed in the nucleus of each cell. If we zoom into the nucleus of an iris cell, there is a specific sequence of DNA that houses instructions for building melanin proteins. Therefore, there are 3 stages of transcription: initiation, elongation, and termination. And just like with DNA replication and transcription, you need to unwind DNA. The difference here is that you only need to unwind a portion of DNA needed for a given trait. So instead of the entire DNA molecule. In this case, we are going to be building melanin proteins to express brown eyes. The area of DNA that will be transcribed is called the transcription bubble. This is made by an RNA polymerase molecule bonding to a DNA in what we call a promoter region, which is upstream from the DNA needed to transcribe. And once the target DNA is found, RNA polymerase separates the hydrogen bonds holding the 2 strands together. Even though there are 2 strands of DNA, only one of them is actually used to make mRNA. The strand of DNA that is read by RNA polymerase is called the template strand, and the opposite strand is called the non-template strand. In the end, mRNA is almost an exact copy of the non-template strand. The only difference is that mRNA has uracil in place of thymine. During elongation, RNA polymerase reads each nucleotide base on the template strand and produces a complement mRNA strand. As RNA polymerase moves down DNA, it unwinds the DNA and then rezips the DNA behind it. Once a DNA sequence is transcribed onto mRNA, RNA polymerase is then signaled to remove itself from DNA, and this leaves us with mRNA containing instructions to build proteins. In our specific case, instructions for building melanin proteins to darken a child's eyes. In eukaryotes, mRNA that is produced by transcription is not complete without some further processing. Even though important genetic material is on the mRNA, there is also a lot of structural filler that needs to be removed. In addition, mRNA is easily destroyed by cellular enzymes and must be coated in protective proteins to prevent this destruction. On the 5' end of the mRNA, a 5' cap is added so that a ribosome can identify this and begin translation. On the 3' end of the mRNA is a long series of adenine nucleotides which protect the mRNA from cytoplasmic enzymes. These enzymes are important in destroying foreign, mainly viral DNA and RNA, from infecting your cells. In addition, there is a lot of non-protein coding sequences in mRNA. Exons are portions of the mRNA that will later be used to build amino acids, while introns are non-coding portions of that mRNA. A process called splicing removes the introns from mRNA and combines the exons so that mRNA is ready to be translated by the ribosome. And now that mRNA has been transcribed from a specific sequence of DNA, it needs to leave the nucleus and find a ribosome so that the amino acids that make melanin. What was that? Hold on. Oh my God! What are you doing?